In part 2 of lecture 16, we want to talk, as we said, a little bit more about regulation of the final volume and solute concentration of urine. So we're talking about regulating the osmolarity and the volume of urine. This material, once again, is from chapter 25 in Merib. Now, let's talk a little bit about where we've been and what we're going to look at in this lecture. You've seen this slide before. This is an overview of the nephron and the functions of the different parts, as we talked about earlier. Remember that we started here at the glomerular capsule and the glomerular capillaries. Remember, we called this the renal corpuscle. And this is going to provide the raw materials for nephron processing. This is the starting point. The fluid that's in here, remember, we refer to as filtrate because it's been filtered through the filtration membrane. The next segment of everything is the proximal convoluted tubule. And we talked extensively about the proximal convoluted tubule, how it works, the primary active transport pump, which is a sodium-potassium pump, which is the mechanism by which it reabsorbs most of what it reabsorbs. The other thing we talked about, remember, was that 65% of the volume of the filtrate gets reabsorbed here in the proximal convoluted tubule. The fluid then moves on, of course, to the nephron loop, which we're going to talk about much more now. And then we go to the distal convoluted tubule, and we talked a little bit about what the distal convoluted tubule does in terms of secretion. Remember, we had sodium ions being reabsorbed in exchange for either potassium or hydrogen ions in the distal convoluted tubule. Now, one of the things we're going to talk about more extensively today is this portion of the kidney, which is here. These are the collecting ducts. And remember, collecting ducts are not formally part of any one nephron. What they do is they receive input from multiple nephrons, as we mentioned before. But the important thing about the collecting ducts is that these provide the option of reclaiming water or letting it pass out of the body. In other words, these play a pivotal role in regulating the final volume and solute concentration of the urine. Now let me just talk a little bit about something, since we're going to be talking about solute concentration, and let me call your attention to this number here, 300 milliosmoles per liter. This is the normal osmolarity in round numbers of body fluids, plasma, and all the other fluids that we have in the body, which we're going to talk about more extensively when we cover water, electrolytes, acid-base balance. But this 300 milliosmoles per liter is a good round number for you to remember. This is the solute concentration of body fluids. Now, if the osmolarity of the body is greater than 300 milliosmoles per liter, that means solutions are more concentrated. If it's less than 300 milliosmoles per liter, that means it's less concentrated or more dilute. So either way, we can look at that. Now, notice that we have this osmolarity in the proximal convoluted tubule. And this osmolarity pretty much will not change in the proximal convoluted tubule. The reason for that is water is free to flow either way from the proximal convoluted tubule into the interstitium or back. So in other words, if the solute concentration changes in one place or the other, water will move into the more concentrated solution and dilute everything out so that we have approximately 300 milliosmoles per liter. This concept of water having to follow solute and always dilute it so that it comes out to about 300 milliosmoles per liter is an important concept known as obligatory water reabsorption, as I have down in the bottom of this slide. So this means that water is obligated to follow solute. There are some areas in the nephron, as we'll talk about in a little bit, where water cannot follow solute. And we're going to have a different term for that, and we'll talk about that in a little while. Okay, so let's talk about the other side of the coin here. We talked about obligatory water reabsorption, for example, in the proximal convoluted tube, where water must follow solute. There are some places in the kidney, and in fact what we're going to talk about right now, for example, right here in the collecting duct, where we have what's called facultative water reabsorption. And what this means is that the hormone ADH, antidiuretic hormone that's secreted by the posterior pituitary, is responsible for reabsorbing water from the collecting duct only when it's present. If you remember, the function of antidiuretic hormone, of course, was to maintain water in the body and keep it from being excreted in the urine. So under the influence of ADH, what happens is any of the fluid that goes into the collecting duct, and I'm on the right-hand panel here of this slide, any of the water that comes through the collecting duct is actually pulled out of that and then can go here out into the interstitium. Eventually this will get reabsorbed into the peritubular capillaries that we talked about in the last lecture. Now, what happens when ADH is not present? Well, when ADH is not there, what happens is the water that's inside these tubules stays inside these tubules and passes right outside the body. So here we have a great mechanism for the body deciding on how dilute or how concentrated the urine will be based on its secretion of ADH. So if, for example, the brain, the hypothalamus, was to detect that body fluids are becoming too concentrated, what would happen is we would increase secretion of ADH. That would increase water reabsorption here from the collecting tubule. This would bring water back into the body, and we would no longer have the problem. This would basically conserve water for us. And as we talked about with the endocrine system, we have a couple of other mechanisms as well that can conserve water and also help us to bring new water into the body via the mechanism of thirst. Okay, so this is basically an overview. Now, I want to talk about an important concept here. Once again, you'll notice I've labeled this graphic with increasing concentration going from the cortical area up here down to the medulla. In other words, the concentration of all fluids is increasing as we go down toward the medulla. We've seen this before. Notice also that in the nephron, the coloration of these tubes basically increases, although it's hard to see here, from a lighter yellow to a darker yellow as we go down here, and then from a darker yellow it gets lighter yellow up here again, and stays light yellow as the water passes out of the body because this is a dilute fluid in here. So once again, I want to call your attention to the fact that we're going to be using graphics that actually key the concentration of body fluids to the color of what we're seeing on these slides, okay? So notice what we call the fluid that's down here in the interstitium of the medulla. We say that this is a hypertonic interstitial fluid. So in other words, this has a concentration greater than 300 milliosmoles per liter. And if the concentration is greater than 300 milliosmoles per liter, that means any solution that has a concentration less than that will have water pulled out of it into the more concentrated solution. The other thing we have is processing of urea, which we're going to come back to. And urea is basically here to help maintain the concentration that we have down here in this hypertonic interstitial fluid down in the medulla. The question that we have to ask ourselves now is, how does it get this way in the first place? How does this interstitium down in the medulla of the kidney get so concentrated that it actually can pull water out of these tubes and bring them back to the peritubular capillaries? That's really what we want to talk about here. Just to make sure that you understand what we talk about when we talk about facultative reabsorption of water, it's the reabsorption of water but only under the influence of ADH. When ADH is present, water can leave the collecting tube or collecting duct and be reclaimed by the body. This is what we term facultative water reabsorption. Okay, so now we want to move on and talk about why the interstitium of the medulla is so concentrated. What gets it that way in the first place? Well, to talk about that, we need to turn to the nephron loop, and this is also called, as we said earlier, the loop of Henle. And this consists of a thin descending limb of the nephron loop, which is right here, and it also consists of a thick ascending limb here. Now, we use the terms descending to refer to fluids that are moving down toward the medulla. We use the term ascending to refer to fluids that are moving back up toward the cortex. So you see by the arrows here that this fluid is moving down toward the medulla, so we call this the thin descending limb. Very importantly, this thin descending limb, as it says, is permeable to water. And we mentioned this before, it's permeable to water but not to salt. So we cannot take any salt out of this tube. The only thing we can pull out of here is water. And so what happens is the fluid, as you see by the coloration, as we move down toward the medulla, is becoming more and more and more concentrated. This is because we're leaving more salt behind in the tube and extracting the water from it. Now, if we turn our attention to the thick ascending limb of the nephron loop, you'll notice these pumps in the membrane of the tubule cells that surround this portion of the nephron. These are sodium chloride pumps, and what they will do is they will pump sodium chloride out of the tubular fluid, which is here, and move it into the interstitium. Now, one of the things we mentioned before and is indicated by this thick black line in this diagram is the fact that this is not permeable to water. So water is stuck in the tube right here and it cannot get out. So what's happening is if we extract sodium chloride from this tube, leave the water behind, notice the coloration of this is becoming lighter and lighter and lighter until we get to the top of this tube and we reach a osmolarity of 100 milliosmoles per liter. Now, if you understood what I said before about normal body fluids being 300 milliosmoles per liter, you'll realize that this is a hypotonic solution at 100 milliosmoles per liter. And in fact, it's important for you to remember that this area right here is where we have the most dilute tubular fluid of any place in the nephron. And that's going to become important because this fluid is on its way later to the collecting tubule or collecting duct. And this is going to provide the basis for us to be able to withdraw water from the collecting duct or collecting tube and pull it back in the body. That's why we want such a dilute solution here, so that we have lots of water to pump into the collecting tube, and then we have the option of whether or not to reabsorb that. This whole mechanism that we see here is known as what's called the counter current multiplier. There's also blood vessels down here, which are called vasorecta, that we talked about before, that are involved in this as well. And this looks like it's accomplishing nothing when you look at this whole picture, but in fact what this is doing is maintaining a concentration of fluid down here in the interstitium that's greater than normal body fluids. And that allows water to be pulled out here. And once again, the reason we do that and how we accomplish that is because of these sodium chloride pumps that are present in the thick ascending limb. The more sodium chloride we pump out, the more water leaves this tubule. The more concentrated this becomes when it goes into the thick ascending limb, the more sodium chloride gets pumped out. So these pumps are responsive to the concentration of sodium chloride inside the tubule. The more salt there is, the harder they pump. So this is what we could consider a positive feedback loop. Okay, so let's take a look now at a text version of this just to kind of review the mechanisms that we just talked about. Remember once again that all movement of water occurs passively by osmosis. That is, water follows solute or water follows salt. And you can see that on the bottom of this slide. Now, what we would like is we'd like some kind of mechanism to be able to concentrate the urine when we want to pull water back into the body, or we would like to be able to lose a lot of water when body fluids are becoming too dilute. And that is exactly the job of this counter current multiplier that we just looked at. And we utilize two factors to do that, as you see right here. We have a hypertonicity of the peritubular fluid down in the interstitium of the medulla. This is established by the counter current multiplier. The other factor is the variable permeability of the collecting ducts to water depending on levels of ADH. Remember, what we called that type of water reabsorption was facultative water reabsorption. Okay, so let's put all this together and take a look at exactly what this accomplishes. This is the same slide that we began with. Now notice that in the case where the body fluids are very dilute and we do not need to reabsorb any water, we will not have any ADH secreted by the posterior pituitary or lower amounts of ADH. What that will do is, because there's no ADH here, there's no water that can escape these tubes. It becomes trapped in these tubes and flows out of the body. So with no ADH or just a little bit of ADH, we excrete a large volume of very dilute urine, which is exactly what we need. We've got to get rid of this body water so that body fluids can be maintained at approximately 300 milliosmoles per liter. Now, what about the opposite case? You're exercising heavily, for example, or you haven't had much water to drink all day. So your body fluids now are becoming more concentrated because, as you realize, just because you stop drinking doesn't mean you stop urinating. So we're still losing water from the body, and if we don't replenish that water with water from the outside, the body fluids will gradually become more and more and more concentrated. So if the hypothalamus receives that signal that body fluids are becoming more concentrated, ADH will be secreted. ADH will affect the collecting duct tubule cells, and what this will do is it will allow water to escape from this tube, and as you see by the coloration here, the fluid is becoming darker and darker and darker. Under the influence of ADH, especially when we have a lot of ADH, we excrete a very small amount of very concentrated urine. I'm sure you've experienced this on days when you haven't had a lot of water to drink. And notice once again that in order to do this, what we have to do is we have to provide a very, very dilute fluid here at the beginning of the distal convoluted tubule. And as we said before, the mechanism to do that was the counter current multiplier that we have down here. So by virtue of the counter current multiplier, we send a very dilute fluid up to the distal convoluted tubule and then onto the collecting duct. And under the influence of ADH, we have lots of water to reabsorb because we have such a dilute fluid that's coming into the collecting tubule. I hope you're getting to understand what we're talking about here. So as it says on top here, note that the amount of water pulled out of the collecting duct is dependent upon the osmotic gradient in the medulla that was established initially by the countercurrent multiplier. That's the purpose of this countercurrent multiplier. Now the other aspect of maintaining a salty medulla in the kidney is the blood flow. I want you to think about something for a second. Imagine if a blood vessel was to start, for example, down here and then just come back up here like this. So let's just imagine that these blood vessels are straight and they're going this way. Well, we know that the fluid in the blood is about 300 milliosmoles per liter, normal body osmolarity. The fluid here is higher than 300 milliosmoles per liter. That means that this is a hypertonic solution. Think about what would happen to the plasma water if it's passing through here and it just made one straight pass through here. The water that's in this blood would leave to dilute the hypertonic medium down in the medulla. And by the time this got up here, basically all of this salt concentration in the medulla would be lost. So basically by the time the blood reached the cortex up here, the water that came from the blood would have diluted all of the interstitium down in the medulla of the kidney. That wouldn't do very well because now we can't produce that very dilute fluid at the top of the ascending limb, and so we wouldn't have the option to reclaim that water later on. So instead, what happens is we have these blood vessels set up that we refer to as the vasa recta. Remember, these are associated with juxtamedullary nephrons, and they have a twofold job. One is to deliver blood to the medullary cells. Remember, these tubules are cells just like any other cells in the body. And the cells that compose these tubules need nutrients, they need oxygen, they need all the same things that other cells in the body need, so they have to have blood. So one of the functions of the vasa recta is to deliver blood to these cells. The other function, very important function in terms of what we're talking about here, is to maintain the salty interstitium down in the medulla of the kidney. In other words, we don't want to disrupt that concentration gradient that the countercurrent multiplier here has worked so hard to set up. And you can basically see the salt flow in here. Salt will flow into the blood vessels at one point, and then salt will flow out of the blood vessels at another point. And the reason for this is take a look at the way the blood vessels are set up. The flow of blood goes down and then comes back up the other side. In other words, this is not just a straight pass as we talked about before, but because the blood vessels are going down and back up, it gives the salt concentration an opportunity to equilibrate with the blood and the interstitium and keep the salt down here in the interstitium of the kidney. So the vasa recta is also a very important part of maintaining the salty medullary interstitium. Now, a couple of other things we want to talk about here, and that is urea and uric acid. Urea is a product of amino acid catabolism. And if you remember the process of deamination, as we said before, remember that we have on each amino acid, we have an amino group. So we have something like this as far as amino acids go. And this portion right over here was the amino group. And during the process of deamination, we took that off. The rest of this molecule then could be used for things like energy or other things that we need in the body. But remember that during the process of deamination, what happens is we wind up with molecules like this, we wind up with molecules like this. So this is ammonia or ammonium ion, and these are very, very toxic to the body. And so what we have to do is we have to convert these toxic products to something that's water soluble, and that is what urea is. So this is a water soluble, harmless substance that can eventually be excreted in the urine. And this is an important number for you to remember. 50% of the urea is reabsorbed, 50% is excreted. If you remember from the diagrams that we just looked at previously, urea is actually used in a cycle down deep in the countercurrent multiplier to maintain the salty interstitium of the kidney. So it actually has a function down there as well. The other substance we want to talk about here is called uric acid. Uric acid is a product of nucleic acid metabolism, so DNA, RNA. And its processing in the kidney is a little bit strange, and it really is still a mystery to me. I've never been quite able to figure out what's going on with this. But 100% of the filtered uric acid is reabsorbed. In other words, all of the uric acid that goes into the filtrate eventually gets reabsorbed someplace in the nephron. And then about 10% is secreted and excreted. So basically we're reabsorbing about 90% of all the uric acid that makes it into the filtrate. And the numbers here basically just tell you how that 90% is broken down. So 100% of it is reabsorbed, and then 10% is secreted and excreted. This slide shows an overall summary of all of the events that we talked about. So this basically puts everything together for you. And on the right-hand panel, as I'm going to go through in a second, you'll see numbered items that correspond to the numbers that are over here in the diagram. So let's take a quick tour through this. You'll notice, first of all, that up here we have the cortex. This is where we have the proximal convoluted tubule and distal convoluted tubule. Down here, of course, we have the medulla. Notice the coloration in the slide. Once again, we're becoming more concentrated as we go down toward the medulla. So we use the same kind of indication in color that we've been using before. Inside the tubules, you'll notice that each of these gray spheres that we see is a molecule of solute. And notice that it's becoming more and more concentrated as we go down the descending limb, and less concentrated as we go up the ascending limb, as we talked about earlier. Now, one of the things we mentioned is that normal body fluids maintain an osmolarity of about 300 milliosmoles per liter. This is normal body osmolarity. This is a nice round number. What happens, remember, in the proximal convoluted tubule is we have something going on called obligatory water reabsorption. In other words, the proximal convoluted tubule is completely permeable to water, and water will always follow salt. Okay, and that's in the proximal convoluted tubule. Now, the other thing we talked about was the fact that aldosterone, which is a hormone secreted by the adrenal cortex, can affect salt reabsorption in the distal convoluted tubule. And remember that when aldosterone is secreted, we reabsorb sodium, so we bring sodium back into the body, but in exchange for that, we either secrete potassium or hydrogen ion, depending on which we have more to get rid of in the body. And then the other thing that we talked about just a little while ago was the ADH regulated permeability of the collecting tubule or collecting duct. And remember, we referred to this as facultative water reabsorption. Okay, so let's go through some of the basics here. Number one is right here, and this is our filtration step. Blood is coming through the afferent arteriole into the glomerulus and is being squeezed out through the filtration membrane, as we talked about in the last lecture, and then leaves the glomerulus via the efferent arteriole. And this is eventually going to form the peritubular capillaries. And remember that in the vicinity of those regions where we have juxtamedullary nephrons, the peritubular capillaries that parallel the nephron loops are known as vasa recta. So filtrate is produced. Remember that this provides the raw material for processing throughout the nephron. And also keep in mind that at the proximal convoluted tubule, we reabsorb 65% of the filtrate. So the majority of reabsorption happens in the proximal convoluted tubule. That's important for you to keep in mind. The next thing that happens is obligatory reabsorption of water. That happens up here. And it also actually happens down here, although this is kind of a special case of it. In other words, water has got to follow salt in these areas. It has no choice. And then we have reabsorption of sodium and chloride by active transport. Now we're talking about number 4, which is right over here. And this is in the thick ascending limb of the nephron loop. Remember, we have sodium chloride transporters, but this particular segment of the nephron is one area where water cannot follow solute. It's blocked from leaving the tubule. So what happens in essence is we're removing sodium chloride from the solution. That yields a much more dilute solution at the top of the ascending limb, the beginning of the distal convoluted tubule, and that's going to give us the option of whether or not to reabsorb this water or let it pass out of the body. And that leads us to the last step here, basically, which is number 6. Under the influence, remember, of ADH, water can be reabsorbed from the collecting tubule, and we can take that water, put it back into the peritubular capillaries, and bring it back into the body. Alternatively, if we don't have any ADH, what will happen is that water will pass right out of the body, and we excrete a large volume of very dilute urine. Okay, and finally we mentioned the vasa recta. Remember, the vasa recta is a system that will not only provide nutrition to the tubular cells in the kidney, but it will also preserve the sodium chloride gradient that we have down in the interstitium of the medulla. And finally, we want to just talk a little bit about diuretics. What is a diuretic? Well, we know from talking about antidiuretic hormone that a diuretic is something that promotes loss of water in the urine. So if you take a diuretic, what will happen is you pee a lot, right? You lose a lot of body water. And there are several different types of diuretics, and I want to mention these because these go along with the physiology that we've been talking about. The first class is known as an osmotic diuretic at the top. And as the name implies, basically if we put a solute someplace, water will follow. That's the whole basis of an osmotic diuretic. We can take something like mannitol, for example, which isn't processed by the nephron at all, And we can put that in the blood, and the mannitol will be filtered out of the plasma, pass through the nephron, and it will not be reabsorbed. And what happens is, because there's more solute in the form of mannitol in the tubular fluid, that draws water into the tubule, and so more water gets locked into the tubule and more water will be excreted in the urine. So this is how an osmotic diuretic works. Now, in the case of diabetics, one thing that happens is, remember we talked about a transport maximum for something like glucose. If there's too much glucose in the plasma, and that eventually gets filtered and processed in the nephron, that glucose has basically overrun the capability of the tubular cells to reabsorb it, and so it will stay in the tubular fluid. It won't get reabsorbed. Glucose acts just like mannitol did. It's a solute. And so what that will do is pull water into the tubule, hold it in there, and eventually will excrete more water along with the glucose. This is why diabetics, as we talked about earlier, have what we refer to as polyuria, because of the glucose in their urine. The second class of diuretics are drugs that, for example, can block sodium chloride transport. As you imagine, what can happen is, if we block sodium chloride transport, we disturb that whole gradient that happens around the medulla, and also in the proximal convoluted tubule and distal convoluted tubule, and this one especially, right? So drugs like hydrochlorothiazide, for example. The another class of diuretics is what are known as high ceiling or loop diuretics. These are very, very powerful, because what they do is they reduce the gradient along the nephron loop down in the countercurrent multiplier. And if we disturb that mechanism, we wind up not being able to take water back in the body, and so we excrete a large volume of very dilute urine. Drugs like Lasix, for example, a furosemide, this can actually cause a very powerful type diuretic, and one of the most common diuretics that we see today. We also have aldosterone blocking agents like spironolactone. These are called potassium sparing. And if you think about this a little bit, if you remember in the distal convoluted tubule, let's indicate this here as a DCT, we have the sodium that's in the distal convoluted tubule. Under the influence of aldosterone, what we have is we have sodium basically being reclaimed into the body, and in return, in exchange for that, we have either potassium or hydrogen ion secreted and then excreted. Well, think about what would happen if we have an aldosterone blocking agent that will block reabsorption of sodium. That means that this won't happen either. And the potassium basically that's in the body will stay in the body. So this is why we call these potassium sparing type diuretics. This is a consideration if somebody has a low potassium. Then we have ACE inhibitors, which are, remember, angiotensin converting enzyme, primarily in the lung capillaries, that basically created angiotensin II, which had all the effects that we talked about before. And so this can actually cause a diuresis because it can interfere with ADH secretion. Captopril is an example of that. And then finally, we have drugs that have a diuretic side effect. And we call these drugs, for example, alcohol, caffeine. Alcohol, if you remember, we talked about earlier, this is one of the things that basically inhibits the secretion of ADH. And if we inhibit the secretion of ADH, we know that water is going to pass right through the collecting tubule and come out. That creates a diuresis. Caffeine is something that actually dilates the afferent arterioles, brings more fluid into the glomerulus, increases the net filtration pressure, and causes more glomerular filtration to take place. And that's the way caffeine causes a diuresis. Okay, so that is Lecture 16, Part 2. In the last part of Lecture 16, we're going to continue and talk about the urine elimination system. So we're back to a little bit of anatomy in the last lecture. I'll see you then.